Government of Telangana. Lab Technician, Grade 2. General Recruitment. Applications are invited from qualified persons online on board's website https://mhsrb.telangan.gov.in for the posts of Lab Technician Grade 2 under various departments of Government of Telangana. Online application will be enabled from September 21st, 2024. The last date for submission of online application is 5 p.m. on October 5th, 2024. Applicants can edit their applications between 10 a.m. on October 7th, 2024 to 5 p.m. on October 8th, 2024. The date of examination is November 10th, 2024. Applications submitted only online will be accepted. The details of vacancies and scale of pay are as follows. Postcode, department, number of vacancies, scale of pay. 1. Director of Public Health and Family Welfare. Director of Medical Education, 1,088, 32,800, 10 to 96,890. 2. Telangana Vaidya Vidhana Parishad, 183, 32,800, 10 to 96,890. 5. M&J Institute of Oncology and Regional Cancer Center, 13, 31,040 to 92,050, 1,284. The zone-wise breakup of vacancies is given at Annexure I. Selection procedure. GO, Ms. number 59, HMNFW, B, Deptanch, dated June 7, 2022. GO, Ms. number 89, HMNFW, B, Depten, dated July 12, 2023. Applicants will be selected based on 100 points, of which a maximum of 80 points will be awarded for the percentage of marks obtained in the written examination, which is a computer-based test. A maximum of 20 points will be awarded for service in state government hospitals, institutions, or programs on a contract or outsourced basis. Educational qualifications. Applicants must possess the following qualification as on the date of the notification. I. Must possess any of the following qualifications. A. Certificate in Laboratory Technician course. B. MLT, VOC. Intermediate. MLT Vocational. With 1 year clinical training. Apprenticeship training. C. Diploma in Medical Lab Technician course. DMLT. D. Bachelor of Science in Medical Laboratory Technology or Master of Science in Medical Laboratory Technology. E. Diploma in Medical Lab, Clinical Pathology. Technician course. F. Bachelor in Medical Laboratory Technology, BMLT. G. Postgraduate Diploma in Medical Laboratory Technology. H. Postgraduate Diploma in Clinical Biochemistry. I. Bachelor of Science in Microbiology or Master of Science in Microbiology. J. Master of Science in Medical Biochemistry. K. Master of Science in Clinical Microbiology. L. Master of Science in Biochemistry. 2. Must be registered with Telangana Paramedical Board. Applicants can register with Telangana Paramedical Board as on date of their application and should upload this certificate online. Age. The applicants should possess a minimum age of 18 years and cannot exceed a maximum age of 46 years. The age is reckoned as on the 1st of July, 2024, as per Rule 12, Subsection 1, Clause A, Item Fee of the State and Subordinate Service Rules. Age relaxations. The upper age limit prescribed above is, however, relaxed in the following cases. Serial number 1. Category of applicants. Telangana. State government employees, excluding employees of TSRTC, corporations, and municipalities. Relaxation of age permissible. Up to 5 years based on the length of regular service. Serial number 2. Category of applicants. Ex-servicemen. Relaxation of age permissible. 3 years and the length of service rendered in the armed forces. Serial number 3. Category of applicants. NCC, who have worked as instructor in NCC. Relaxation of age permissible. 3 years and the length of service rendered in the NCC. Serial number 4 category of applicants: SC, ST, BCs and EWS. Relaxation of age permissible: 5 years. Serial number 5. Category of applicants: physically handicapped persons. Relaxation of age permissible: 10 years. The examination will be a computer-based test and will have 80 multiple-choice questions, with each question carrying 1 mark. The computer-based test will be held on the 10th of November 2024. Annex your I. Breakup of provisional vacancy position: S. Nomen Department, Zone Y, Zone 2, Zone 3, Zone 4, Zone Phi, Zone 6, Zone 7. Total: 1. Director of Public Health and Family Welfare or Director of Medical Education. 187, 111, 145, 159, 137, 180, 169. Total: 1088. 2. Telangana Vaidya Vidhana Parishad. 31, 24, 28, 32, 12, 40, 16. Total: 183. 3. MNJ Institute of Oncology and Regional Cancer Center. 13. Total: 218, 135, 173, 191, 149, 220, 185. Total: 1,284. These posts are not under the purview of presidential order. Annex your 5. Syllabus for examination: diploma level. Clinical biochemistry. Unit 1. General topics. Laboratory services. Levels of laboratories: primary level, secondary level, and tertiary level. Reference laboratories. Infrastructure in the laboratories. Laboratory space includes reception, specimen collection, quality water supply, power supply, work area, specimen or sample or slide storage, cold storage, record room, and fire safety. Personnel in the laboratory. Qualifications as per NABL document. Equipment, listing, cleaning, maintenance, SOP, verification of performance, internal quality control. Instruments, theoretical basis, uses, different parts, installation of instruments like calorimeters, spectrophotometers, balances, centrifuges, refrigerators, hot air ovens, water baths, thermometers, vortex mixers, magnetic stirrers, UV lamp, and spectroscopes. Reagents and materials, purchase, maintenance, storage, and use. Specimen collection, storage, and transport, general guidelines of sample collection, labeling, handling, transportation, storage of specimen. Care in handling specimens. Pre-analytical considerations including patient preparation. IP or OP numbers and LABI numbers. Worksheets, reporting test results, specimen rejection record, recording of laboratory data, maintenance of records. Types and mechanisms of action of various anticoagulants and preservatives used. Serum and plasma preparations. Preparation of protein-free filtrate. Sample stability. Water, chemicals, solutions, and reagents. Types of water. Characteristics and principles of preparation of distilled and deionized water. Distillation apparatus and deionizers. Chemicals. Different grades of chemicals and their significance. Solutions. Definition and preparation of percent, normal, molar, and molal solutions. Concept of standard solutions. Principles of preparation of standard solutions. Primary and secondary standards. Unit 2. Units of measurement. Different systems followed and their interconversions. Colorimetry. Principles. Complete understanding of percent transmittance, absorbance, and Beer-Lambert's law. Concept of using blank, standard, and test, B, S, and T. Mathematical calculation of quantity of an analyte through formulation of general formula. Electrochemistry, basic principles and applications of potentiometry. Ion selective electrodes. accidents and emergencies, and first aid in the laboratory. Bio-waste management. Automation basic principles, different components, and general principles of usage applications. Ethical considerations. Transmission of ethical values, voluntariness, compliance. Computers. Basic theoretical knowledge and usage of computers in clinical biochemistry laboratory. Quality assurance. Standard operating procedures, SOPs. Definition, format. Internal and external quality assessment. Introduction. Unit 3. Biochemistry. Chemistry of carbohydrates and reactions of glucose. Carbohydrate metabolism, glycolysis, citric acid cycle, HMP shunt, oral glucose tolerance test in normal and diabetes mellitus, ketone bodies. Chemistry of lipids and lipoproteins, chemistry and metabolism of proteins. Ammonia metabolism, urea cycle, chemistry of nucleic acids, structure of DNA, RNA, ATP, GTP, UTP. Non-protein nitrogen substances, NPN, urea, creatinine, uric acid. Digestion and absorption of carbohydrates, lipids and proteins, vitamins and minerals. Unit 4. Clinical biochemistry. Acids, bases and salts. Concepts of hydrogen ion concentration. pH and pH meter. Plasma proteins, fractionation techniques, interpretation. Bilirubin, outlines of formation and excretion. Conjugated and unconjugated bilirubin and its clinical applications. Jaundice. Urine formation and characteristics of normal urine. Abnormal constituents. Urine analysis and interpretation. Spot urinary protein, 24 hours urinary protein. Renal clearance tests and total protein and Ag ratio. Blood urea, serum creatinine, BUN. Body fluids, outlines of formation, composition and analysis of body fluids like cerebrospinal fluid, pleural fluid and peritoneal fluid, and interpretation of the results. Glucose tolerance test. Concepts of tolerance tests. Definition. Patient preparation, performance, reporting and interpretation of GTT. Blood coagulation, basic knowledge. Principles of estimation of plasma fibrinogen and prothrombin time, APTT. Lipids and lipoproteins, triglycerides, cholesterol, phospholipids. Serum lipoprotein, patterns in health and disease. Enzymes and isoenzymes, LDH, CPK, GGT, SGOT, SGPT. Hormones, analysis of 17 ketosteroids and VMA. Inorganic ions, concepts. Bicarbonates, phosphates, calcium, sodium, potassium. Blood gases. Analysis bicarbonate buffering system. Henderson-Hasselbalch reaction. Blood pH, PO2, PCO2, HCO3, calculations. Nomograms. Unit 5. Demonstration topics. Liver function tests, gastric analysis, renal function tests, radioisotopic techniques, calculi formation and analysis, thyroid function tests, electrophoresis. Clinical microbiology. Unit 1. General microbiology. Knowledge of various samples and procedures of collection for microscopy, serology, and culture. Procedures of accepting or rejecting samples for cultures. Procedures of labeling, registering, transport, and storage of various specimens. Procedures of laboratory work, safety, and laboratory maintenance. Knowledge and expertise in working under conditions of biological hazard, BSL 1, 2, 3. Procedure of handling exposure to various infectious agents. Biomedical waste management. Maintenance of laboratory equipment. Culture methods for different bacteria and fungi. Media preparation. Methods of inoculation. Disposal of used media and specimens. Sterilization and disinfection. Laboratory and operation theaters. Unit 2. Bacteriology. Characterization of aerobic and anaerobic bacteria. Differences between aerobic and anaerobic. Grams staining method. Reagents preparation. Albert's staining, reagents preparation. AFB staining, reagents preparation. Characterization of gram-positive bacteria. Cocci, Staphylococcus and Streptococcus. Bacilli, Mycobacteria, Clostridia and Corynebacterium. Gram-negative bacteria. E. coli, Klebsiella, Proteus, Salmonella, Shigella, Vibrio, Pseudomonas. Principles of bacterial identification. Staining, culture, spot tests, commercial kits. Unit 3. Mycology and parasitology. Knowledge of common pathogenic fungi. Differences between yeasts and molds. Fungal media preparation, pH adjustment, preservation of media and samples, preparation of reagents. KOH, germ tube test, spot tests. Knowledge of various parasites and diseases caused by them. Life cycles, morphology, hosts, vector, animal reservoir, epidemiology, parasite, host relationships, diagnosis and treatment. E. Histolytica, Giardia, Cryptosporidium, Ascaris, Echinococcosis, Schistosoma, Toxoplasma. Blood parasites, Plasmodium microfilaria. Light microscopy, reagent preparation, staining methods. Normal saline and Lugol iodine mounts. Serological methods for parasite detection, identification of antigens in blood, feces, and urine. Unit 4. Virology. Knowledge of medically important viruses. General laboratory tests for viral detection. Viral serology and ELISA. Tests based on immunochromatography. Maintenance of viral laboratory. Unit 5. Automation in microbial identification. Automation in bacteriology. Blood culture systems. Methods of antimicrobial sensitivity tests. Disk diffusion test. Kirby-Bauer, MIC, E-test. Principle of action of antibiotic agents. Detection of multi-drug resistant, MDR, bacteria, ESBL, MRSA, VRE, Mycobacterium. Serological tests for antigen and antibody detection. Pathology. Unit 1. Clinical pathology. Urine collection, physical and chemical examination. Sugars, proteins, ketone bodies, Benzojones proteins, bile, blood in urine. Microscopic examination, formed elements, casts, crystals, parasites, abnormal cells. Other body fluids, types, names, physical and chemical examination. Semen analysis, physical, chemical and microscopic examination. Unit 2. Hematology. Knowledge about RBCs, WBCs, platelets and any abnormal cells. Hemoglobin estimation, red cell indices, hematocrit, ESR. Total cell counts, WBC, RBC, platelets, AEC. LE cell test, coagulation-related tests. Hemoparasites, bone marrow slide preparation. Cell counter operation. Unit 3. Histopathology, knowledge of material and equipment used. Collection, preservation, and labeling of samples. Grossing methods, fixatives used, and processing of tissue, including bone tissue from collection to slide making. Special stains and immunohistochemistry. Staining procedures. Preservation of specimen, blocks, reports. Mounting of museum specimens. Unit 4. Cytology and general pathology. Different types of techniques and equipment used to obtain materials, including various guided methods. FNAC, imprint smear, vaginal smear, buccal smear, cytospin cytosmears. Various staining methods. Different reagents used in pathology. Preparation of fluids for cytological examination. Transport of specimen. Preservation of samples, reports, records. Disposal of waste, infected materials. Unit 5. Immunohematology. Material and methods used in blood banking. Blood grouping techniques, ABO system and RH system. Donor selection. Storage and processing of blood bags. Blood bags. Grouping, quality control test and labeling. Direct and indirect Coombs test, antibody titers. problems in blood grouping and cross-matching.